Hey there, it's Dr. Beevler again. We're going to finish out the mitochondrial heteroplasmy discussion with a couple of other pathology or pathologic conditions that can be that has been shown to be associated with mitochondrial heteroplasmy, and those would be neuropsychiatric diseases and cancer. So, neuropsychiatric diseases are very diverse. That would include Alzheimer's disease, Parkinson's disease. Some of the papers talk about Down syndrome and other kind of more common psychiatric illnesses such as depression, anxiety, etc. So in this particular paper, it talks about that the mitochondrial genome encompasses over 1,000 DNA genes, plus hundreds of thousands of copies of maternally inherited mitochondrial DNA, and therefore partial defects in either nuclear DNA or mitochondrial DNA, or in combination of the two, can be sufficient to cause neuropsychiatric disorders. Inherited and acquired, and we're more interested in the acquired somatic mutations, mitochondrial DNA mutations, has recently been associated with autism spectrum disorder, which parallels previous evidence of mitochondrial DNA variation in other neurologic diseases. And therefore, mitochondrial dysfunction may be central to the etiology or the cause of a wide spectrum of neurological diseases. And this paper is talking particularly about Alzheimer's disease and Parkinson's disease, and I'll read some of this. So, somatic mitochondrial DNA mutations are elevated in Alzheimer's disease, Parkinson's disease, and Down syndrome and dementia, both in brains and systematically. Alzheimer's disease, Down syndrome, and Down syndrome with Alzheimer's disease brains also have reduced mitochondrial mRNA levels, altered mitochondrial copy number, and perturbed A beta metabolism. And basically the conclusion is that therefore mitochondrial dysfunction provides a unifying genetic and pathophysiologic explanation for Alzheimer's disease, Parkinson's disease, and other neurodegenerative diseases. If we go over to the right here, we can see that on the top picture is what is considered basal conditions or healthy neurons. And basically what you're going to see is that they just are more organized. The mitochondrial networks are more diverse. There's more mitochondria in general, which is going to provide more energy for the cell, and it's going to function much better. And then if you look at the potential pathologic conditions, Alzheimer's, Parkinson's, et cetera, the mitochondrial networks are not they're not ordered. The mitochondria are smaller and less elongated, and as well as the mitochondria are less plentiful. And then a little detail that you may not be able to see because the legend's covered up by my face is that the healthy neurons have more autophagy going on, and the pathogenic conditions have less autophagy going on, which we'll talk about in much greater detail during the mitochondrial dynamics section. So the last thing I wanted to cover was basically that cancer is also included in the conditions that are associated with mitochondrial heteroplasmy. And this paper, it talks about notably many recent studies have reported a heteroplasmy shifting phenomenon as a potential shaper in tumor progression and treatment response. And we suggest that each cancer type has a unique mitochondrial heteroplasmy shifting profile. So mitochondrial heteroplasmy is going to be different for every single type of cancer, but it's definitely associated with cancer, and it's something we definitely want to be looking out for. To conclude, I just want to go over kind of a paper that was written by Doug Wallace, one of the premier mitochondrial researchers, and it's titled "A Mitochondrial Paradigm for Metabolic and Degenerative Diseases, Aging and Cancer: A Dawn for Evolutionary Medicine." And what the highlighted portion, it just says, since the mitochondria use oxidative phosphorylation to convert dietary calories into usable energy, generating reactive oxygen species, ROS, as a toxic byproduct, I hypothesize that mitochondrial dysfunction plays a central role in a wide range of age-related disorders and various forms of cancer. And hopefully during this mitochondrial heteroplasmy series, we're able to see that there pretty much every problem that people go to doctors for or suffer from as a human being can be related to mitochondrial function as well as mitochondrial heteroplasmy, which determines its function. And because mitochondrial DNA is present in thousands of copies per cell and includes for essential genes for energy production, I propose that a delayed onset of progressive course for the age-related disease result from the accumulation of somatic mutations in the mitochondrial DNA of post-mitotic tissues. So basically, majority of the diseases we see. Now, are there inherited mitochondrial conditions? Yes, but they're fairly rare. But the majority of the problems that we see with people these days, whether it be high blood pressure, whether it be cholesterol, whether it be obesity, whether it be diabetes, whether it be heart disease, heart failure, neurodegenerative diseases such as Parkinson's, Alzheimer's disease, and cancer, are going to be a result of somatic mutations in the mitochondrial DNA after birth by epigenetic causes, which we're going to talk about in great detail. But the vast majority of that damage is caused by reactive oxygen species, a toxic but necessary byproduct of energy metabolism. And that's what this series is about. It's about mitochondrial redox, and we're going to figure out how to actually make sure your redox is balanced, and that way you aren't causing excess damage to these mitochondria, which then causes premature aging and the diseases that no one wants. So with that, I hope that we have covered mitochondrial heteroplasmy well. If we have not, please, put in the comments, your questions, or maybe something that's still fuzzy. But hopefully that we have, made the case that not just mitochondrial dysfunction, but the precursor to that, which would be mitochondrial heteroplasmy, which then leads to mitochondrial dysfunction, is what is leading to a lot of these diseases that no one wants to have. So with that, I will wrap up, and we will be going next into mitochondrial dynamics, which will be several video series as well, and then it starts to get a little bit more exciting. So with that, till next time.